Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speakers your questions. Your questions in the Q&A window will only be visible to myself and our speakers. So thank you for joining us today. It's now my pleasure to introduce our speakers, Prasanna Srinivasan, PhD, Research Scientist, Center for Biomedical Innovation, MIT, and Graziella Piras, PhD, Senior Director, Strategic Marketing from 908 Devices.
Thank you very much for the introduction, Alex. And hello, everyone, and thank you for joining us on this webinar today. I would like to give a special thanks to MIT for being engaged with 908 Devices in this great collaboration. And a very special thank you also to Prasanna, who is going to present today on his work on developing a process for scalable AAV production. In today's world of the development of new type of therapies, cell and gene therapies, there's an increasing need for more scalable and more efficient and more robust processes. And this is so true in the gene therapy space to produce viral vectors where the titers are still below what they need to be to reach the market needs. And really to develop more efficient processes, we need more understanding of these processes, which in part can be achieved by better real-time analytics. At 908 Devices, we have actually set our mission to develop such analytical devices for upstream bioprocessing. We can see here the Maven and the Maverick, which are two devices that provide online and inline analytics for upstream bioprocesses. We also see here on the left the Rebel, which is an at-line amino acid, vitamins, and other amines analytical device. And this is the device that we will be focusing on today. So a little bit more about the Rebel. The Rebel is an at-line analytical device that can sit right next to your bioreactor. It has a fairly small footprint. And also, the great thing about the Rebel is that you only need about 10 or less microliters of volume to analyze your sample. And you will get basically concentrations of all the amino acids, some vitamins, and bioamines. It is amenable to high throughput. You can either put vials or a 96-well plates, and you will get results in about 10 minutes for each sample. It is very easy to operate, no need for integrating peaks, no need for an analytical PhD scientist. Any bioprocess engineer scientist can operate the Rebel. Therefore, it is very efficient and a cost-efficient method to analyze amino acids at the point of need. So how is the Rebel used? It is used in many different applications shown here, for example, to accelerate media development and feed optimization. It is used in some of our examples in even the CAR T cell process development, and of course in gene therapy optimization and process intensification. So without any further ado, I'm going to hand it over to Prasanna, who's going to present his project.
Thank you, Graziella, for the introduction. I'd like to start by saying I'm just a voice of all these people that you see on the slide. These people have contributed to the different stages of the project I'm going to present today. Today, I'm going to tell a story about how by taking advantage of Rebel measurements, we could improve the scalable AAV vector production for gene therapy application. As most of you know, gene therapy requires delivery of healthy therapeutic gene into patients. This is generally done via two approaches, viral and non-viral approaches. This talk will focus exclusively on viral-mediated gene therapy and in particular adeno-associated virus. AAV is a 24-nanometer nuclear capsid. This capsid is made up of 60 subunits of 3 different types of capsid proteins, shielding a single-stranded DNA cargo. The genetic elements of the cargo are exchangeable to achieve therapeutic gene expression in a tissue-specific manner. Tissue targeting is achieved by changing the composition of the capsid proteins because AAV has evolved to infect variety of tissues. There are about 11 natural serotype and several engineered variants, making it attractive for gene therapy applications. Unlike wild-type AAV genome, gene therapy vector is a replication incompetent, thereby making it safe carrier for therapeutic gene. Industrially, there are two approaches available to produce AAV vectors. One is baculovirus-mediated production using insect cell line, and the other approach is triple plasmid transfection of mammalian cells. Today, I'm going to talk about the triple plasmid transfection of mammalian cells. Triple plasmid transient transfection is a workhorse of AAV production. This process involves using three different types of plasmids: plasmid G, plasmid RC, and plasmid H. Plasmid G codes for therapeutic gene of interest. In our case, it is a GFP reporter gene. Plasmid RC codes for rep proteins and viral capsid proteins, and plasmid H codes for adenovirus genes, which assist in AAV vector production. These plasmids are mixed with cationic transfection reagents, which is polyethanolamine in our case, to form DNA PEI complex that is delivered to suspension HEK293 cells. Typical transfection density is between 1 to 3%. Transfected cells, when harvested, result in the production of both filled and unfilled capsids. The population of the filled and unfilled capsids varies with serotypes, but typically it is in the range of 5 to 20%. Of all the capsids that are produced, only between 5 to 20% of the capsids are filled, and majority of the capsids that are produced are empty. There are various reasons why we produce more empty capsids instead of filled capsids. And AAV production inherently causes a lot of cellular stress because there is a cytotoxic effect associated with the expression of Rep78 and 68 and PEI addition. So current AAV production produces more impurities than the actual product. Product-related impurity removal is critical and challenging. As you can see, there are a variety of impurities associated with a product. Cells can produce empty capsids. It can also produce partially filled capsids and capsids with endogenous DNA fragments. In our presentations, we are going to categorize the capsids are whether it is filled or whether it is empty. But irrespective of how we do that, separation of the capsids filled with the transgenes with the impurities is very challenging. And this put a lot of burden on the downstream purification, increasing a lot of cost and a labor-intensive process. And one of the big challenges is the downstream burden includes a lot of expensive process like chromatographic separation and ultra-centrifugation. Furthermore, scaling of the vector production is also a big challenge in this field. As you can see, irrespective of the vector production volume, the critical product attributes have to remain same, and maintaining the critical product attributes across the production volume is extremely challenging. Furthermore, presence of impurities also causes a lot of undesired immune response to the patients, and it also reduces the efficacy of a dose. So we need an approach to enrich the product upstream in order to reduce the impurity levels, which in turn reduces the cost. We apply data-driven mechanistic models to get an understanding of AAV production process. What you see here is the plot that we got from triple plasmid transient transfection of a 30 ml shake flask culture. The points that you see are obtained from the experiments, and the solid traces are obtained from mechanistic models. The plot that you see on the left shows the dynamics of viral DNA replications. You can clearly see, within the first 24 hours, the number of copies of viral DNA is really very low, and the number of copies started to increase only at the later time points. And this causes a time mismatch between viral DNA replication and the capsid production. Because within the first 24 hours, majority of the capsids are produced, thereby leading to most of the capsids with empty and having very small population of the capsids filled with the viral DNA cargo. So the objective is to reduce the amount of impurities that are formed during the viral vector production process. And the mechanistic models allows us to identify the bottlenecks in the AAV production. Our typical process is like it is produced either using the reactor setup or in a batch shake flask culture. Samples that are drawn from these processes are used to estimate the capsid titer with ELISA and droplet digital PCR for vector genome titer. And this information is directly fed into the mechanistic models, which in turn provides a lot of information to improve the process. Now with the addition of Ribble into our process pipeline, we get an additional information to improve our mechanistic models. So Ribble data informs data-driven mechanistic models to improve the AAV production process. And this is very valuable to reduce the cost of the product by reducing the impurities. Typical sample preparation using Ribble is very straightforward. Like the samples that are obtained from the cell culture are separated from supernatants and cells, and the supernatants are just mixed with the diluents, and these samples are then loaded into the plates which are provided, and that comes along with the kit. And then with the hit of a button, we get a snapshot of the entire panels of amino acids and small molecules that are present in the sample. The sample preparation takes about 5 minutes, and we can repeat that for several samples that are drawn at different time points. And today I'm going to discuss about the 2 case studies where we used Ribble measurements to get an understanding about what are the additional constituents that need to be added in order to improve the vector production process. The first case study involves a batch process where we compare 2 different cultures. The first culture is an untransfected culture, and the second one is a triple plasmid transient transfection culture. So we use triple plasmids with this GFP, RC5, and helper in equimolar concentration at 1 microgram per ml. The DNA PA mass ratio is 1:2. And we tested 2 different media formulations. One is freestyle 293 expression medium from Thermoscientific, and the XL medium from the Millipore Sigma. On the right, you see the cell density and the viability plot that we got from freestyle expression media, comparing the untransfected and transfected conditions. What we see here is actually the growth rate gets reduced beyond 48-hour time points, and we see that both in untransfected and the transfected conditions. And we also see the drop in viability beyond 72-hour time point from 90% to close to about 80%. So there is no appreciable difference both in the cell growth rate between the untransfected and the triple plasmid transient transfection. Next, we compare the amino acid panels between the spent media and the original freestyle media before we started the culture. On the left, what you see is the amino acid concentrations of the freestyle 293 expression medium. What we see here is actually the 20 amino acid concentrations. In particular, we see a very low concentration of glutamine because the media contains glutamax, which is a dipeptide of glutamine and alanine, from which the glutamine can be derived. And the essential hydrophobic amino acids that you see, they are all within the concentration of 1 millimolar. The 4 millimolar serine concentration that you see is very critical because serine is a precursor to the biosynthesis of several amino acids as well as the lipid metabolism. And tyrosine that you see is also obtained from the phenylalanine, and it is conditionally essential. On the right, what you see is actually the depletion of a couple of essential amino acids that we saw from the transfected and the untransfected conditions. And what you see here is like the amino acid concentration monotonously it decreases from the time point 0 to the time point 72. And here we have shown for histidine and phenylalanine for the transfected and untransfected conditions. There are other essential candidates, and they also follow pretty much the same trend as these candidates. But the striking result that we got from the Ribble measurement is actually the relative levels of glutamine and asparagine in transfected and untransfected conditions. So what you see here is two different types of traces. The first trace is the solid trace, and the second trace is a dotted trace. And the dotted trace corresponds to the untransfected conditions, and the solid trace correspond to the transfected conditions. So we can clearly see that the glutamax level drops from the time point 0 to the time point 72 monotonously. And that happens both conditions in transfected and untransfected. But the relative concentrations of glutamine and asparagine for the transfected conditions is higher compared to the untransfected conditions. In particular, at the time point 50, many interesting mechanisms happen. We saw that it takes at least about 48 hours for the cells to break the glutamax and relieve maximum amount of glutamine in extracellular. And furthermore, the capsid production also saturated by that time. And close to about 50 hours, we see maximal viral DNA replications. Because of all this demand that is exerted by the cells, the endogenous glutamine is also added to exogenous glutamines, which is already supplemented in the media, and that results in an increase in the relative concentration. And we see that consistently happening close to 50 hours, and which is really evident from this result where like the glutamine and the asparagine concentrations for the transfected conditions are higher compared to the untransfected conditions. And asparagine in particular is involved in protein synthesis, and AAV-VP3 capsid protein contains 10% of asparagine, and it is a very significant amount, and we see that it is reflected in the Ribble measurement. And this is also further complemented by the capsid titer produced by the cell. Here we plotted the concentrations of the asparagines in the untransfected and the transfected conditions overlaid on the capsid titer. We can clearly see that the capsid titer, as it starts to increase until to the point of 50, the asparagine concentration increases, after which there is a sudden drop in the asparagine concentration in the transfected conditions. In contrast, for the untransfected conditions, it follows a completely different trend. Based on the understanding, we decided to go ahead and further compare whether the presence of glutamine and asparagine in certain proportion is going to improve the AAV vector production. And what you see here is a plot on the left where the capsid titer per cell is plotted for different media compositions. First, we started with the Freestyle 293 media, which already has a glutamax in it, where we got a per cell capsid titer in between 1 and 2 e to the power of 4. And when we supplement the Freestyle media with the extracellular asparagines, we did not see any big change in the capsid titer per cell. Then we used another media, XL, which does not have any glutamax or glutamine added to it, but it has asparagine in it. And again, when XL media is supplemented with the glutamine, we did not see any big change in the capsid titer compared to the previous two conditions. But if the glutamine is deprived from the XL media, we saw roughly about twice the capsid titer increase compared to the earlier conditions. So this result clearly confirms that depriving glutamine in the presence of asparagine increases the capsid titer. And this data has been reported earlier in Cell Metabolism, where they clearly show that glutamine, when it is available in the extracellular environment, it is taken up in the TCA cycle to produce asparagine. Whereas when the glutamine in the extracellular is cut off, the endogenous glutamine is not sufficient enough to produce enough asparagines. So the protein synthesis pathway is abrogated, which led to the growth arrest and apoptosis. Whereas After cutting off the extracellular glutamine, if you're going to supplement with an asparagine, that is enough for the cell to once again grow and proliferate. And we see somewhat a similar mechanism even here. So based on this understanding, we try to go ahead with the next step of understanding AAV vector production for the next case study. And this involves an alkaline process analytics for a continuous AAV production. And in this experiment, what we did is like we used a 300 ml Applicon bioreactor with Artemis VHF filter for perfusion, and we used a triple plasmid transient transfection to transfect the HEK293 cells. And the process was monitored for 11 continuous days. The bolus that was added at different time points were obtained from the mechanistic models. And we also obtained the perfusion rate based on the mechanistic models. The plot that you see on the right bottom tells you the total capsid titer that is produced continuously during the span of 11 days. You can clearly see that the capsid titer continuously increased over the 11 days as expected, because that's what the bolus was optimized for. Next, we had drawn samples at the different time points to estimate the amino acid concentrations. And what you see on the left is the three-dimensional color plot where each color denotes one amino acids. And we had got amino acid concentration for 11 days for all amino acids. And when we did a cross correlation with the daily capsid production, we had an interesting result here. On the right extreme, what you see is a Pearson correlation coefficient plotted against different amino acid labels. You can clearly see there's a positive correlations for the candidates like beta-alanine, asparagine, and tyrosine, and a negative correlations for candidates like glutamine, glutamate. And in particular, we can clearly see the negative correlation of the glutamine is compensated by the positive correlation of asparagine. And this is quite expected because in the earlier result clearly showed that the asparagine is produced using glutamine pathway, and it is an important part of the capsid protein composition. So in summary, I want to conclude by saying the performance of the host cell machinery is the key for producing the filled capsids, and Ribble system assists in identifying the nutrients necessary for keeping the cells happy. And we also identified the previously discovered glutamine asparagine metabolic pathway, which plays a very critical role in AAV production. And we observed that the capsid titer can be increased when glutamine is absent in egg cell media. So Ribble measurements enable rapid at-line analytics to inform data-driven models. And using these models, we would be able to predict the AAV production. We are doing more Ribble measurements in our subsequent experiments to better understand how amino acid metabolism affects high-density transfection. And this is very important for producing few doses of AAV for ultra-rare diseases. Finally, I would like to thank the leadership team of the CBI for supporting to execute this project. Thanks to Professor Anthony Sinsky, Professor Richard Bratz, and Dr. Stacy Springs. I also would like to thank research staff at MIT CBI for their constructive feedback and suggestions. And I also want to extend my thanks to the 908 Devices collaborators, in particular Mila and Graziella, for coordinating with us at the different stages of the project. And lastly, I would like to thank the funding agency USFDA. Thank you for listening, and I would be happy to take your questions.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speakers now. So our first question is, can you briefly explain how models are applied to batch and continuous production runs?
Yes, like we build models using data-driven approaches, and these data are generated from the in-house experiments, both in batch as well as in semi-batch system. But as far as continuous production is concerned, we don't have a prior data set. So the bolus is determined based on whatever data sets that we have based on batch and semi-batch systems that we had run previously in the bioreactor setup. But as far as the batch runs are concerned, like we have enough data to give a better prediction. Now with the addition of Rebel Analytics, we are trying to improve the model to better predict the capsid titer and improve our process.
All right, great. And our next question is glutamine asparagine effect on capsid titer specific to the AAV serotype produced?
Yes, that's a good question. So as far as capsids are concerned, capsid compositions are concerned, like a majority of the sequences are conserved actually. There are some changes related to some amino acids depending upon the target location where it binds to the tissues and other things. But when we overall compare the serotypes based on the compositions, asparagine plays a very critical role, and we significantly have somewhere between like 8 to 10%. For the AAV5, it is close to 10%, but for rest of them, it varies between some 7 to 10%. So it is really an important component for the AAV production. So having a system like Rebel trying to get an estimate of how the asparagine and glutamine concentrations vary during the production process really helps.
Our next question is, how is data reported back from the Rebel and what type of software is needed?
So I can answer that. Yeah, the data from, so first of all, you get the data in millimolar. So every analyte is reported back in millimolar, as in your starting material. And you can export the data very easily as it comes into, it shows on your screen, but also it's exported in a CSV file. So you can export it in Excel. It can also be seamlessly exported in the JUMP software with very nice add-on to the JUMP software for visualization. So you can compare conditions, you can compare bioreactors for very easy analysis. And then from the CSV file, of course, into your data analytics, statistical analysis tools, it is a very seamless export.
All right, great, thank you. Does amino acid depletion lead to recombinant AAV product quality issues such as misincorporations in the capsid proteins?
Exactly. Yeah. Depletion of certain essential candidates which are required for AAV production results in the production of a lot of impurities. Because if you see, both glutamine and ASN both are required at different stages of AAV productions. They are required for the viral genome replication, and they are precursors to nucleobase sugars. They are precursors to some of the essential amino acids which are being produced during the AAV production process. So if you are going to have a concentration level that is less than what is required, we would end up producing more empty capsids than the filled capsids, and which results in a lot of product impurities being produced instead of the actual product.
All right, great. Well, it looks like we're running just a bit short on time. So if you don't get to your questions, we'll be passing them along to Prasanna and Graziella, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, you stated at the beginning that the titer for AAV production was still too low. With your results, how far are you from the targeted titer, let's say, for a commercial process?
Okay, yeah. So we, after doing the optimization of the media supplementation based on glutamine and asparagine pathway, we have got to a situation where we have got like roughly about like threefold greater than whatever we used to get before with the actual media formulation that was commercially procured. But there is a lot of opportunities and scopes to improve and optimize this process. And I would anticipate like at least you would be able to get at least an order of magnitude greater. So yeah, currently we are working on developing some new processes based on that.
All right, fantastic. Well, thank you so much, Prasanna and Graziella.
Thank you.
Thank you.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with the direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.